Okay, thank you. So we are going to move on in this particular session on systematics and evolution, and I'm very pleased to be able to announce Dr. Geng Qin. I hope I pronounced your name somewhat right, who is going to move us into looking at those full genome sequences to discuss how genome sequences reveal global dispersal routes and convergent genetic adaptations in seahorse evolution. Dr. Jin, are you able to share your screen with us?
Yeah, okay, thank you, Haley. I will show my slides first.
Okay, wonderful, thank you so much.
Oh, thank you. Okay, can you see my slide now?
We see your slides in presenter mode. There you go. Okay, yes.
Okay,
and
right.
We're starting a little bit late, so you have 20 minutes until 8:54.
Oh, it's okay, thank you. Oh, thank you, Haley. Yeah. Okay, hello everyone. My name is Geng, not Geng Qin. Geng from South China Sea Institute of the Oceanology, Chinese Academy of the Sciences. I'm very pleased here to give my presentation with the title Genome Sequence is Reveal Global Dispersal Rules and the Convergent Genetic Adaptation in Seahorse Evolution. I will introduce my I will tell my stories about the following biopaths. Firstly, about the introduction. Although, as we all know that a lot of the factors driving the biodiversification, including the environment and the biological factors. And one of the most key important factors is that the major geological recurrence events. For example, the Tethys Sea, the Panama Sea, and the shallow waters in the Indo-Pacific Sea areas. For example, the previous report had figured that the marine biodiversity hotspot had moved from the west to the east during the last 40 million years ago. And also, a lot of species have been proved that the species expansion occurred allowing the closure of the Tethys Sea. In addition, a series of biology trees related to the lifespan, the dispersal ability, and the ecological niche are also important for the for the marine biology. Affecting their distribution, colonization, genetic diversification, and finally speciation or extinction. Therefore, it's also important to pay attention to the potential effect of the evolution of the adaptive phenotypes when we're trying to figure out the truth about the marine diversification. Okay, so we can talk about the seahorse now. Why I want to pay attention to seahorses? Firstly, because they are in danger. Now, all the seahorses have been listed in the IUCN list, red list, and the seahorses are commonly recognized as the flag species for a wide range of the marine conservation issues. Besides, the seahorses are also kind of iconical and suitable models animals for investigating the dispersal and the diversification of the marine taxons, for they have some unique characters. Firstly, all the seahorses are not all the seahorses are widely distributed around the world except the polar cold areas, and also seahorses have weak swimming abilities because of their unique body shape and the rafting dispersal with a prehensile tail. And also seahorses are less of pelagic periods because of their male pregnancy. What's more is that among the different species of the seahorse is the body size, color patterns, and the bony spines and so on varied a lot. So they make the seahorse is also a good chance is for us to study the adaptive trees for the marine biology in response to their local environment. Besides, also the seahorse genomes evolve under some of the highest mutation rates among the tier list and have the greatest diversification rate within the family. That means that there might be much more mutations could be provided for the seahorse is for developing adaptive trees for diversification or adaptation. In fact, about the mechanisms of the seahorse diversification and colonization is all have been a hot topic in the last 20 years. There's a series of good papers and excellent scientists, including the Professor Peter Tisk and the Steve Casey. They have done a lot of work about this topic. So we could easily find a series of good hypotheses and the evidence. For example, we all believe that the tectonic events such as the Texas Sea and the ocean currents play some kind of key roles in the seahorse long distance dispersal and the global colonization. However, there are also still some confusing or discrediting problems that we need to find more evidence based on more data. Luckily, with the faster development of the new technology, we now have the chance to study this hot topic based on the whole genome sequences. The genome sequences will give us more credible phylogenetic trees. The whole genome also could help us to get better divergent times and the gene flow details, and also provide the chances to investigate the genetic basis for those adaptive biology traits. So in the past years, about the 2015 to 2017, we tried to get as many as samples about the seahorse all around the world. Luckily, we finally got about 358 specimens comprising 21 species, which covering almost all the corners of the world and also covering the major lineages according to the previous reports. And we resequenced all the specimens and get data at an average about 16-fold coverage. In addition, we also tried to using the PacBio and the high-seq technology to get a new advanced genome of the Hippocampus erectus. And the high-quality genome will help us to find more details about the genetics basis about the biology trees. Then use the data bio, we performed the following analysis. Firstly, we get a strong NG trees based on all the SMP data of all the specimens. And the results showed that the seahorse are monophyletic, and there are remarkable different clades in line with their distribution. For example, the big belly seahorse is branched off first, and then two major clades are recovered, while the clade 1, the red and the green one, only inhabiting the Indo-Pacific regions, while the clade 2, including the brown and the blue lineages, they covering almost all around the world, not only the Indo-Pacific region, but also the Atlantic oceans. Interestingly, we find that even in the Atlantic Ocean, the South group and the North group separate from each other significantly. What's more, we analyzed the genetic diversity, and we also found that the North Atlantic groups with a higher seed values compared to the South Atlantic groups. So why is the phenomenon? So we try to use we try to get a phylogenetic trees and the divergent times based the calibration points, including some seahorse fossil information. And the results showed that the common ancestor of the all extended seahorse species live about 20 to 25 million years ago. And then there are two remarkable speciation waves at 20 to 15 million years ago and 9 to 3 million years ago. Interestingly is that we can easily find that the seahorse is living in the North Atlantic areas and the South Atlantic areas. They are separated from their sister lineage just at the two waves respectively. So further, we try to do something more to answer these questions. The topology and the branch lengths of the species trees were used to reconstruct the geography diversification and to different models of the diversification in space. And then we find that, firstly, the Indo-Australian archipelago, just the IAA areas, was identified as the center of the origin of the seahorse genus. And subsequently, the seahorse diversified and spread globally. For example, the clade 1 lineages, they inhabiting the IA areas and around that, while the clade 2 lineage spreads westwards to the East Africa. As that, before the 50 million years ago, the Texas Sea is still open. The ancestor seahorses of the North Atlantic lineage have a chance to disperse across the opening Texas Sea into the Atlantic Oceans. And again, with the help of the ocean currents in the Atlantic Oceans, the seahorse is dispersed further into the Gulf of Mexico. And then after the Texas Sea closure, the seahorse began to spread southwards along the East Africa coastlines with the help of the ocean currents. Finally, they reached to the Cape of the Good Hope, and further passing around the Good Hope into the South Atlantic Ocean areas. Similar, with the help of the ocean currents, the seahorse is dispersed across the wild Atlantic Oceans and to the brazier, and then going across the opening Panama Sea into the East Pacific Ocean. There are some also other evidence proving our hypothesis. First is about the effective population size. We could find that the effective population size of the ancestor seahorses of the North Atlantic land age, the value is really small, that most probably due to the closure of Texas Sea. And another evidence is the living seahorse species have complex captains. These small species are only living in the tip of the South Africa, which is belonging to the land age of the South Atlantic group. So another interesting point is about the East Pacific group. Just refer to the hippocampus engines. According to the above results, it seems that the hippocampus engines were divided from the ancestor from the IAA areas and dispersed westward straight crossing the south of the South Africa and also the Panama Sea, and finally arriving the East Pacific areas. This way is much different from the previous reports. That the previous report said that most of the marine species from the IAA areas across the wild Pacific areas by the North Way. In addition, we also calculated the gene flows among different species. We found that most of the gene flow were detected between the species inhabiting the South Atlantic Ocean groups. That may be because of these species are all younger than the others. And also, this may indicate that this seahorse is still on the way of the colonization and even the speciation. And also, we could still we could find the evidence of the seahorse is moving back from the East Africa to the IAA areas, suggesting that the global dispersing contributing to also could contribute to the high species diversity in the Indo-Australian archipelago areas. And then, as we talk something about the adaptive phenotypes evolution, here we pay attention to the spanning trees. The spanning trees were found in 4 seahorses among the 21 species. And the 4 seahorse species all have this kind of the diamond bony spines, which are an effective defense against the predator. But the 4 species seem to evolve independently across the species tree. So to investigate the molecular basis of these repeated evolved adaptive phenotypes, we performed a positive selection analysis and identified 37 genes with significant singles in Spanish seahorses. Among these genes, we focused to the BMP3 genes. We found that the BMP3 genes were reported to be involved in bone development by affecting the ostabulate diffusion in mammals. And the BMP3 protein tree showed different from the species tree, and the 4 Spanish seahorse learn it exhibits independent amino acid changes either. And again, we try to check the positional function. And the hormone-instituted hybridization demonstrate the specific BMP3 expression domains in the seahorse spines during their early development. And moreover, a knockout experiment using the CRISPR-Cas9 in zebrafish showed that the mutant have a series of significant skill effects, such as the decrement in skill numbers, rearrangements, and irregular shapes. So these results suggesting that BMP3 might play a role in the formation of the spines in seahorses. Okay, let's go get out the take-home message now. Firstly, the global diversification of the seahorse involved long-distance dispersal and has been facilitated by the Paleo-CV dynamics and changing ocean currents. In the Pacific, seahorse colonized the Atlantic Ocean by two distinct routes and in 2 waves. The third is about the genetic basis of the recurring bonibines adaptive phenotype is linked to independent substitution in a key development gene, BMP3. All about the work have been published in the Nature Communications in this early year. So if I didn't tell the details, you can find them in this paper. Finally, I want to firstly thank to the Linchang's members. And that's our members. Especially, I want to thank my partners, Li Chunyan, who is also the co-first author of these papers. In addition, I want to thank our collaborators from the Aximia's lab, the Lucian May's lab, and the Golden Xia's lab. I thank Melissa, thanks Ralph, and thanks Dr. Hoya Li. Okay, thank you, everyone. That's my presentation.
Yes, thank you. I know that we are all applauding electronically for what seems to me like 3 talks in one. What a fascinating set of questions you've asked using some really interesting molecular approaches. We already have a couple of questions that are here in the chat. So Jin is interested to understand if the chromosome level genome of Hippocampus erectus is now publicly accessible.
Sorry, can you say it again? I didn't get that.
Yes, if the chromosome level genome of H. erectus is publicly accessible.
Yeah. Also.
Excellent. That's excellent. If there's a reference you want to drop in the chat, perhaps that would be great, so that we can respond to that particular question. And there's time, you can put it in the chat whenever you want. But so that's great. Josephine, would you like to ask your question directly?
Sure. Thanks for your talk. First of all, I really enjoyed that. I was wondering, so your age estimates for the different seahorse clades are quite different from what is already published by Peter Teske and colleagues. Do you know if that has any impact on those dispersal routes you reconstructed, and how did you decide to place the fossils in the tree?
Thanks, Taylor's question. In fact, we have discussed our results with the previous reports, including the Peter Tix and also the Stephen Cassie's papers. Yeah, in fact, most of the trees is similar, but it's true that there are some remarkable difference, and so far we prefer to believe that our results is more strong. But I still to impress that most of our opinions is based on the results of the scientists, just what I mentioned. I want to thank them. I don't know whether I give a proper answer.
Well, you certainly have much more taxon sampling than the Teskey paper did back then. There's another question, a really interesting one from Tony Wilson that's in the chat. Just for the sake of time, because we're running a little behind, Jin, if you would be willing to answer Tony's question in the chat so we could possibly move on to our next talk and make sure we have time for our coffee break. So I'm very pleased to, so thank you very much for that comprehensive and fascinating look into the evolution of hippocampus, biogeography, spines. We really appreciate it.